Barriers come in a lot of different forms. The fences affecting pronghorn migration, the virus keeping us socially distant, and in my personal experience, this very cheeky bull elephant, separating me and my guide Simon from our field vehicle. While some barriers are obvious, others exist as a result of our complex world, hidden from sight but impactful nonetheless. For my MEM project, I was able to focus specifically on barriers, and not always the elephant kind. My name is Melissa Butynski, and during my time with the MEM program, I was a distance student living abroad in Kenya. With a background in wildlife biology and over 4 years of professional experience, I was able to use my project to contribute to an ecosystem-wide study on the genetic connectivity of African lions. With less than 2,000 lions currently left in Kenya, this iconic species is quickly disappearing. The exponential increase of human population, coupled with modification of the land for urban, energy, and agricultural development, has severed the connectivity of the landscape. Barriers, both seen and unseen, separate populations, confining wildlife to areas which are too small and too far removed from others to allow for natural processes and genetic movement to occur. The African lion is not only a vital component of the ecosystem, but also a predominant draw for the tourism industry, which accounts for nearly 10% of Kenya's gross GDP. Lions require vast space for hunting and breeding, often bringing them within close proximity to humans. Conflict, ignorance, and misconceptions of their important predatory roles in the ecosystem have all contributed to population decline. Conservation of this iconic species for ecotourism and ecological value is a key goal of the Kenya Wildlife Service's 50-year strategic plan, with the goal to restore and maintain robust and connected populations of lion and their prey, while minimizing conflict with local communities. To contribute to this effort by the National Wildlife Service, a Yale University PhD candidate named Mary Burak began a collaborative project with local lion conservation organization Lion Landscapes to define the genetic corridors of lion in the Ewaso ecosystem, which is located in central Kenya. The Ewaso ecosystem boasts a wide variety of land uses, from communal lands which support livestock grazing and small-scale agriculture, to large private entities focused on wildlife conservation and tourism. Only 5% of the ecosystem is formally protected, with 1% dedicated to private wildlife reserves. The variety of land use and human densities across the landscape offer the perfect setting to examine how human presence and land management practices can affect wildlife conservation as our rapidly expanding urban environments force people and wildlife into more frequent contact. It's important to note that many of the local communities are also well aware of the importance of an intact and functioning ecosystem and are generally inclined to find a way to coexist with species. Partnering with multiple private landowners and tribal communities, this project aimed to collect genetic data throughout the ecosystem. This novel information is critical to establishing and protecting wildlife corridors and helping local organizations, governments, and communities create comprehensive land management plans which emphasize coexistence between people and wildlife within the Ewaso ecosystem. My role was to act primarily as the project manager during the duration of data collection from May 2019 to January 2020. Working as a key communicator and a coordinator for the project, I was able to provide field assistance and ensure the project continued to run smoothly while Mary was out of the country. Being incredibly familiar with the project area, I was able to hit the ground running and begin collecting data from multiple properties in the region. Genetic data was collected through non-invasive Scat Collection, which happened within 10 wildlife-friendly private ranches. Working closely with local biologists and guides, we found that the best method for finding fresh, high-quality scat was to visit recent kill sites. Armed with my little red bucket, silica beads, and a collection of vials, a typical day would involve a drive to the project site, some of them being up to 4 hours away from where I was staying. I would meet with the local biologist, or the property would assign a wildlife ranger to help me find a recent kill sites that had been reported either by GPS collar locations or visual sightings from rangers and visitors. Once a recent kill site was found, we would spend as long as necessary, combing through the surrounding area for any recent scat. In addition to collecting the scat itself, we would also take note of habitat, prey species, and as well as a detailed account of human activity, including livestock, housing, roads, and other man made structures. For Scat Collection, we focused on collecting the outside of the feces. This would contain the genetic data of that individual lion. This individual genetic data would allow us a more in depth understanding of gene flow within the area. We will be able to use this information to identify genetic corridors, or lack thereof, and provide this information to local management agencies so that they can decide how to best create a coexistent landscape which balances the needs of wildlife with those communities which frequently come into conflict with these predators. Obviously, safety was always a key factor during data collection. Many of these kills had happened within a day or so, which meant that the lions would remain nearby, feeding until the carcass was gone. We also had to remain vigilant for additional wildlife in the area, such as elephant, hyena, and certainly the creature that makes me most nervous, the Cape buffalo. Working closely with our local guides, they were able to make sure everyone was safe and proper precautions were taken. During the data collection portion of this project, we were able to collect 209 usable individual scats from 10 of the participating wildlife ranches within the ecosystem. As this study covered such a large area, some of the ranchers had been trained on scat collection themselves and stored any samples for us until we could return to pick them up. It was truly a collaborative effort, and it was amazing to see so many properties, organizations, and individuals come together for this research effort. As for next steps within the project, thankfully all data was collected by the end of February. Genetic analysis had been set to begin in March, but unfortunately, the COVID-19 crisis shut those plans down. Mary will continue with the genetics portion as soon as she's able to get back to Kenya, but as many other things in life, it remains to be seen when that will happen. I plan to continue collaborating with Mary where I can. We are currently in the beginning of a side project literature review, looking at the connections between invasive plant species and wildlife connectivity. I'm also thankful to say that I will be co-authored on some of the future publications that will come from this genetic study. This project gave me the opportunity to continue exploring my interests in wildlife connectivity and large-scale conservation efforts. I'm thankful to Mary for taking me on as a field assistant and trusting me with something important as her PhD project. While I may not be able to be directly involved with the remainder of this landscape genetics project, I am happy to report that I will be able to continue working on similar projects in East Africa and abroad after graduation. I will be joining the Center for Large Landscape Conservation, which focuses on community resilience, science and research, policy and law, as well as facilitating collaboration. I'd like to give a quick thank you to first and foremost Mary Burak for adding to her workload and taking on an eager master's student from beyond her own institution. And of course I have to thank Jessica Young for being a guiding voice, an ear to bend, and a champion of distance students. And finally, I would like to thank the Lildaiga Hills Research Program. This project was only possible thanks to their generosity in providing housing and logistical support.